Hello viewers, in the ongoing series on lipids, I have discussed compound lipids in my last few lectures by taking examples of glycophospholipids in detail, and I have also introduced you to sphingolipids. In the present lecture, we will first revisit sphingolipids, and I shall also be discussing with you sphingomyelin, which is an example of sphingophospholipid, and another class of lipids, that is glycolipids. So in my last lecture, we had discussed glycerophospholipids, and I had introduced you to sphingolipids. So let us see what is the difference between glycerophospholipids and sphingolipids. The major difference lies in the alcohol, which makes the backbone of these lipids, that is sphingolipids and glycerolipids. As you can see on your screens, I have shared with you the structure of sphingol, which is also known as sphingosine and glycerol. Now, glycerol is the backbone of glycerolipids, and we know it is a trihydroxy alcohol, whereas sphingol is a amino alcohol, aminodiol. So the difference lies in the backbone. In sphingolipids, the backbone alcohol molecule is sphingol, and what we find in this is that this molecule has an amino group along with two hydroxyl groups, and it has a very long side chain, which is unsaturated. And if we talk of glycerol, then in glycerol we have three hydroxyl groups. So when we have three hydroxyl groups, what we find that in glycerol all these three hydroxyl groups are active. That is, they'll make a bond with acid residues, carboxylic acids, resulting in formation of triacyl esters, that is oils or fats. And if they react with two molecules of carboxylic acids and one molecule of phosphate, they will result in phosphoglycerolipids. Whereas when we talk of sphingol, then what we find in sphingol is that the first hydroxyl group, which we take from the bottom, and the amino group is on the second carbon. These two groups are biologically active. That is, they'll take part in a reaction, that is, a bond formation will take place here. Whereas the third OH is not biologically important, that is, it does not take part in the reaction. So here again you can look into the structure of sphingolipids, and what we find in sphingolipids, that sphingosine is the backbone of this molecule. And when we talk of sphingosine, as you can see here, I have not shown a OH bond here. This is a diagrammatic representation. And what you find, why is the OH not shown here? Because as I said, that this OH is not biologically important. So we tend to diffuse it. But please remember, it is present here. And this is the long chain fatty acid, unsaturated fatty acid. So whenever you see this structure in the molecule, it should immediately come to your mind that this is a sphingolipid. Now here we have the amino group here undergoes reaction with fatty acid, resulting in a amide linkage here. And now the main difference in all the sphingolipids will be at this point. In this case, in case of sphingolipids, what we find that the OH at the first carbon will undergo a phosphoester bond with phosphate, resulting in the phosphosphingolipids, and then here we can have a nitrogenous base, which can be an amino alcohol. And this is the polar head of the molecule. Whereas when we talk of glycerophospholipids, what we find that two of the hydroxyl groups are involved in ester formation with fatty acids, that is long chain carboxylic acids, and the third OH is involved in the phosphoester bond and the alcohol molecule. So what is the difference between the sphingolipids and the glycolipids, glycerophospholipids or glycerolipids? The main difference is the backbone. That is, in sphingospholipids, the backbone alcohol is sphingosine, whereas in glycerophospholipids, the backbone alcohol is glycerol. So this is what you have to keep in mind, the basic difference between a sphingolipid and a glycerolipid is the difference in their alcohols, which make the main skeleton of the molecule. Now let us discuss sphingolipids. Like, what are sphingolipids? As I've already said, that they are a class of lipids that contain a backbone of a set of aliphatic amino alcohols, one of which may be sphingosine. So that means these lipids contain alcohol sphingosine, a fatty acid and a polar group. So that the central alcohol molecule in sphingolipids is sphingol or sphingosine. When we are talking of sphingol or sphingosine, we are talking of the same compound. And when we talk of sphingosine, what we find, as you can see on your screens, that it is a 18 carbon molecule alcohol, and it has and we start the numbering from here, please remember, because we are going to number it according to lower sum rule. So this is 1, the amino group is having number 2, and this is the third OH. And then when we go down, what we find that the double bond is between the third and the fourth carbon. So in this case, what we find that this OH group and this amino group is biologically important, and these are the groups which will take part in the reactions resulting in ceramide or derived phospholipids. So now let us go on to the next form. So that means sphingosine will form the primary part of sphingolipids, and these sphingosine is a class of cell membrane lipids, right? So when we talk of sphingolipids, what we find that in sphingolipids, the hydrophobic region is made up of long chain sphingoid base such as sphingosine, which is linked to the acyl group of a fatty acid via an amide bond. So if you look into the structure, what we find that if this is the structure of sphingosine, this is the structure of sphingosine, what we'll find is that this amino group will react with a fatty acid and a amide linkage will be formed here. And this OH group will also take part in the reaction. Now see, when we talk of sphingolipids, it may be sphingosine or it may be glycolipids, which we'll be discussing also. What we find is that this molecule, this basic structure is going to be the same in all sphingolipids. Where will the difference lie? It will lie in this group H. If this group H is H as in sphingol, then what we find that here it will depend on the nature of this polar group here. That is, what will be the nature of the phospholipid that we will get, or what will be whether we'll get a phospholipid or we'll get a glycolipid. So when we talk here, we will get a amide linkage here, and this will be your OH is the polar group. So this part of the molecule is hydrophobic, whereas this part, which is your polar part, we will modify the molecule sphingol from here, and what we'll find that this polar part, when which is attached to it will depend on the group R which we replace at this H, that what kind of sphingolipid are we getting. So now here I have shown you the general structure of a sphingolipid. So what we find that this is the long hydrocarbon chain which is unsaturated, and we'll have a acyl group here, and then we have a hydroxyl group which is not taking part in the reaction. And then we have here, this is the long chain hydrocarbon, here you have a acyl group. This is the one where we are getting a amide linkage. Here the NH2 group which is present in sphingol reacts with the acid group and we get a acyl group here. So we will get a amide linkage here. Now this R will vary in different sphingolipids. It can be H or it can be some other group. So this is the basic structure of a sphingolipid. So when we talk of a ceramide, ceramide are the simplest sphingolipids. As I said in my previous slide, I have discussed with you that depending on the nature of the R1, we will get different sphingolipids. And as I have said, ceramide is the simplest lipid. And what we find in this, that the R group is in the ceramide is a hydrogen atom. So that means what is a ceramide? Ceramide is a fatty acid linked to an amino group of sphingosine with an amide linkage. So if you look into this structure, so what we find that this is the first carbon here, which is attached to OH. That is, we've not modified, we've not replaced this H of sphingol with any other group. And this OH does not take part in the reaction. As I told you, this is not very active. The amino group at the second carbon reacts with a long chain fatty acid, resulting in a amide linkage here. So this is the basic structure of ceramide. And what we have to remember, this basic structure will be present in all sphingolipids. So now what is ceramide? So if you don't remember the structure, what you have to remember is that ceramide is a sphingosine, and what you have to remember, at the second carbon you are going to make a amide linkage, and that will give you a ceramide. So what is a ceramide? Ceramide is sphingol plus fatty acid. Please remember, the fatty acid is at the second carbon, that is at the amide bond. So this is what is a ceramide. That is, this is your first carbon. Here H is retained from the sphingol, and this amino group at the second carbon goes acylation, resulting in the formation of a amide bond here. So this is your ceramide. And you have to remember that ceramide is the simplest sphingolipid. All other lipids are going to be derivatives of ceramides. And this ceramide is widely distributed in small amounts in plants and animal tissues. And as I've said, all other sphingolipids are derivative of sphingosine, of ceramide, So what you have to remember here is this is the basic structure which is going to be present in all sphingolipids. So you don't have to get confused. So just remember the structure of sphingosine and you can make any sphingolipid from here. That is the basic structure you have to remember is the inverted L, and this, of course, here we will have a amide bond, and this here the fatty acids may differ. And when we are making the derivative, what you have to remember is that this H is going to become our R1. That is, if we replace this H with some other groups, we will get other classes of sphingolipids. So on the basis of the group R1, sphingolipids can be divided into two subgroups. One is sphingofospholipids and the other is sphingoglycolipids. Now what you have to remember is when we classify them into two groups, that when we are talking of sphingofospholipids, here the active group or the polar group here is going to be the phospho group. And in this category, we are going to discuss today in this lecture sphingomyelin. And when we talk of sphingoglycolipids, what you have to remember is that here the polar group is going to be carbohydrates. Please remember, glyco refers to the word carbohydrates. So when we are talking of sphingoglycolipids, what you have to remember that the backbone molecule is sphingosine, whether we talk of sphingophospholipids or sphingoglycolipids. Here, the active group or the polar group is going to be phosphate group, and here it is going to be carbohydrates. For sphingophospholipid, we shall be discussing sphingomyelins, and for sphingoglycolipids, we shall be studying cerebrosides, gangliosides, and gangliosides. So, the common polar group R, as I said, may be phosphate. In that case, your sphingolipid will be sphingophospholipid. And if the R group is, say, a single monomer unit, or it is having two sugar molecules, or it has some nitrogenous base in it, we can have glycolipids. And these glycolipids can be of three types. We can have cerebrosides, globosides, and gangliosides. So that means now if we look into the basic difference in the two structures of sphingophospholipid and glycolipids, what we find in that both the molecules, this sphingosine residue is going to be there. This still here. And then where will the difference lie? That in sphingophospholipid, we will have a phosphoester. That is, this OH will be attached to a phosphoric acid, resulting in a ester bond here, and we will have a nitrogenous base, and we will have a amino alcohol here, which will make the head of the molecule. And in glycolipids, we will have a carbohydrate residue here, which may be just one molecule, or it may be more than one molecule, that is maybe 2 or 3, resulting in globosides, or it may be containing some complex oligosaccharides. So when we say that if a phosphate group is attached to an alcohol group, and generally this alcohol group is choline, it will give us sphingomyelin. So what you find here on your screens again, that the basic unit is the same here, and what has been done, that the R group here has become this phosphoryl choline. That is, the linkage is with phospho group, and this phosphate group is then making a bond with choline. So what we get here is sphingomyelin. And the next thing that you have to remember is that if in the molecule, now if I go back to glycolipids, and if the R group is a single sugar unit, it will give us cerebroside. So here you can see an example of a glycolipid on your screens, and what you find here, that the R1 group here is a glucose molecule, and what we get here is a glycolipid. This could be glucose, this could be galactose, or this could be 2 or 3 sugar molecules here. When we'll get globosides, we can vary the sugars here when we will get a globoside. So depending upon the structure of the R group, we will either get sphingophospholipid or we will get a sphingoglycolipid. If there is a phosphoester bond, we will get a sphingophospholipid, and if there is a bond between the ceramide and a carbohydrate unit, then we will get a glycolipid. Now let us see what is sphingomyelin. So when a ceramide is linked to a phosphate and a nitrogenous group, it will give rise to sphingomyelin. So what you find here, that this is the basic structure of a ceramide. This is the basic structure. That is, we have a OH group at this position, then we have a amide group here, and then this OH group is involved in phosphate ester bond. That is, we get a phosphoester bond, and then this phosphate is making a bond with an amino alcohol when we are getting sphingomyelin. Now these sphingomyelins are the most important sphingophospholipid. And as you can remember, you don't have to mug up. What you have to remember is sphingomyelin is made up of ceramide, and the H of ceramide of the first OH group is replaced by a phosphoryl group and a nitrogenous base. It will result in sphingomyelin. And you already know that ceramide is a sphingosine and a long chain fatty acid. So now in sphingomyelins, the phosphoamino alcohol may be phosphate and a choline, or it may be phosphate and an ethanolamine. So in these cases, we say that sphingomyelin usually consists of a phosphocholine and a ceramide, or it is made up of phosphoethanolamine head and a ceramide. The OH of the ceramide binds to the phosphate ester group of choline to form a sphingomyelin. And this makes, when a phosphate makes a bond with choline, that group is known as phosphoryl choline. That is, phosphoryl choline is phosphoric acid plus choline. So as you can see on your screens now, you can see the structure of sphingomyelin. So when we see the structure of sphingomyelin, what we find, this is the long chain alcohol, that is sphingosine, and the OH here is not involved in bond formation. Amino group at the second position is involved in bond formation with a carboxylic acid, resulting in the formation of the amide bond. And this group, that is the phosphoryl choline group, is the head of sphingomyelin. And this, please remember, is the polar group here. Another example of sphingomyelin is that we can change this head group here, and this head group may be phosphoethanolamine. So if you look at here, that this is the OH, the O group of the ceramide, which is entering into bond formation with phosphoester bond with a phosphate, and then there is a bond between ethanol first carbon, second carbon, and then you have a amino group here. So this is another example of a sphingomyelin. Therefore, since if you look into the structure of sphingomyelin, what we find that in sphingomyelin again we have a phosphate group here. So the sphingomyelins can be rightly called as sphingophospholipid. So what is a sphingomyelin? Sphingomyelin is a type of sphingolipid found in animal cell membranes, and it is one of the principal structural lipids of membranes in nervous tissue. Now, sphingomyelin is found specially in the membranous myelin sheet that surrounds and insulates some nerve cell axons. Therefore, it is also called as sphingomyelin. These compounds play a very important role in signal transmission and cell recognition. So sphingomyelins are very important. And these compounds also play a vital role in regulating the programmed cell death or apoptosis. So now here we've discussed sphingomyelin in great detail, and we know that sphingomyelin is an example of sphingophospholipid. And for this, I had referred to books by Bruce Fowler, McMurray-Jones, Finnar, Morrison and Boyd, and Solomons. In my next lecture, now we shall discuss glycolipids, where I would like to discuss first compare glycolipids with phospholipids, and then from there we'll take the lecture further. Thank you.